there, everybody. We'll go ahead and let folks start coming into the Zoom room now. Thank you so much for joining us for Biology Essentials, Biology and Environmental Science Essentials. We are so excited to explore these sensors with you. While people are kind of coming into the Zoom room, let's kind of get acquainted with one another. I'd love to find out who has joined us for a webinar before. At the bottom of your Zoom window, you should see a chat bubble. If you click that, go ahead and type in the chat box if you've joined us for a webinar before or if this is your first time. Just make sure that that to section is set to everyone. That makes sure that everyone can see your chats. People know what you're talking about. This will be especially helpful at the end of the webinar while we're asking questions or answering folks' questions. All right, it looks like Lisa is joining us for the first time. Hey, Lisa. Anyone else who is new and joining us for the first time? Do we have any, folks that have been to every single webinar? I see Anne. I recognize that name. Thanks for joining us again. Candace has definitely been with us before. Stephen, Matt, awesome. Thank you so much for coming to our webinar today. We are so excited to dive into biology and environmental science essentials with you all. Awesome. Well, it is 3:01 Pacific time and make the most of our time today. I do want to go ahead and jump into some housekeeping items. So we'll pop to the next page. Awesome. Just, for your information, a recording of this webinar will be live on our website and on our YouTube channel in about 3 to 5 business days. We will also send an email with a link to the recording as soon as the recording is edited and ready to go. In the email with the link to the recording, you will also find additional resources and a certificate of attendance. I'll link that in the chat section here in just a second as well, so keep an eye out for that. Furthermore, we'd love to answer your questions during today's session. Please feel free to use the chat section in Zoom to ask your questions. I'm going to keep an eye on it, and if I see any really pertinent questions, I'll stop Melville and kind of ask. Otherwise, we will have a questions and answers section at the end. So if we don't get to you during the webinar, we certainly will get to you at the end. And if we happen to miss your question, we are available. Please email us at biology@vernier.com. All right, I will pass you off to our amazing instructor today. Thanks, everyone.
Hi, everybody. So I'm just going to cover the agenda really briefly. You're going to meet me. You already have met me, but you're going to meet me. And then I'm going to introduce you to what I call the core sensors for biology and environmental science. There's going to be some demonstrations that are going to be integrated with that. So there'll be some live demonstrations, some videos that I'll show you. But there's also this really nice chat function. So if you have questions at any time, please just pop them there and chat. Just make this feel more like we're all sitting together in real life. And if you have questions, just ask them and we'll get to them. And I can just stop and answer any questions that you have. There's many links to online resources that'll be useful later that you'll get, or if you have any questions about where I went to or what resources to get or where on the website, don't be afraid to just email biology@Vernier and ask, and I'll get them to you. All right. So just so you meet me, I think some of you have met me before. My name is John Melville. Everyone calls me Melville, but my first name is John. I got my bachelor's degree in biology and psychology at Sonoma State University. And then I got a PhD in zoology from Oregon State University. I did postdoctoral research at University of Oregon, and then was an associate professor of biology at a little college called Wartburg College. And my big thing that I did is I taught majors and non-majors courses in biology and neuroscience, including marine biology. And I really liked active learning, and I really tried to emphasize data collection and inquiry in all of my courses. So that's really how I got my first experience with Vernier and other data collection platforms. So yeah, the most important thing really though is that I'm a fellow teacher or was a former teacher. That's the most important thing. And so if you have any questions about how to use our equipment in the classroom, what's the best way to use it, or other questions like that, feel free to ask, because that's what I'm here to cover. All right, so what I first wanted to talk to you about though is you may have heard of our Go Direct sensors. Our Go Direct sensors are sensors that are used directly with your phone, tablet, Chromebook, etc. They do not require an interface, so you don't have to buy a separate interface to get them to work. You can use them in two ways. So two ways. You can use them over USB, so they have a USB port and a USB cable, or you can use BLE. BLE stands for Bluetooth Low Energy or just Bluetooth. So you have two ways to use them, Bluetooth or the USB cable. Almost all of them have a really easy calibration process. That's all of the biology ones. Or for some sensors, no calibration at all is required. And the software that you would use them on is graphical analysis. There's a free version of that software that you can use that anybody can get to that's on the website. I can show you that really quickly. If you go to the Vernier website right here, you can go up to the top right corner where it says downloads. And if you go to downloads, you'll see the Direct Sensor companion apps. Here's graphical analysis. If you tap on graphical analysis, you can download it for free. There's also a progressive web app, so I can launch graphical analysis by going to a web address. There's lots of ways to do that. If you have an Android or iOS device, a phone, you can install it there as well. So that's why we call it Go Direct, because it can be used on any platform. Now, it can also be used on LabQuest. So some of you may remember our own interface that has a screen called the LabQuest. This is LabQuest Viewer. So there's a box that looks a lot like this that's called the LabQuest. This is a LabQuest 3. You can also connect Go Direct sensors via Bluetooth or USB to the LabQuest, LabQuest 2 and LabQuest 3. All right. Any questions?
No questions yet. We did have a poll though. Should I launch that?
Yeah, go ahead and launch the poll.
Awesome.
And then we'll come right back. So I believe in the poll, all that we want to do is get a better handle on what do you teach? Is it K through 12 or college? And then what are you most interested in? Are you more interested in the sensors that I've got to show you, or are you more interested in how the software works and different ways to use the software? Just wait for a minute for y'all to vote, and I will get ready to show you some cool things here. Also, I believe Dylan said there's going to be a drawing for a free Go Direct temperature probe. So if you're attending, I believe there's a good chance you could win a Go Direct temperature probe.
That's right. Yeah, we'll do the drawing at the end. So make sure you stick around to find out if you won a free Go Direct temperature probe. I can't wait to get into that.
Great. Okay. That is so the results of the poll are actually kind of exactly what we thought. Most of you probably teach K through 12, but some of you do teach college, and almost all of you are here to learn more about the sensors, which is exactly what this talk is focused on, which is great. Once again, don't be afraid to ask any questions. All right. So what I'm going to do is I just want to show you one of these sensors. So when I say the live demo, what I'm going to do is I'm just going to show you a screen. So here, I think you can see it. This is a Go Direct temperature probe. And here is a Go Direct CO2 sensor. And you see they all look this way. Here's a Go Direct oxygen gas sensor. They all have this nice blue color and gray. That's how you can tell they're a Go Direct sensor. And let me just show you here. I'm going to tip this Go Direct temperature probe up. You can see that right here, that is the USB cable or USB port. So I can plug it in over USB and connect it to graphical analysis. It also has a little button right here. And if I tap on that button, it'll flash. And that means the Bluetooth is broadcasting and you can connect it to graphical analysis that way as well. So I just want to show you how quick and easy this is to do. So let me just show you a side-by-side view here. So here I've got a Go Direct temperature probe, and I've launched graphical analysis over on the left. And I'm just going to take the cable here. I'm going to plug it into my Go Direct temperature probe. You heard it make the nice sound. there's this little blue light here, and what you should be able to see over here is that the sensor has already been connected and everything is ready to go. You can see temperature down here. It's a live readout for temperature. The collect button is live, and if I hit collect, I can then grab onto the temperature probe. I'm just grabbing onto the tip, and you should see that the temperature goes up. So the simplest way to get any of these GoDirect sensors to work is by using the USB port. I'll hit stop. Let me just zoom in there. All right, so what about using them wirelessly? Let me just show you how to use them wirelessly really quick, and then we'll go on to some other sensors. Does anybody have any other questions? I'm just disconnecting the sensor there. I'm going to say new experiment. And when you say start everything over, it'll pop up and say graphical analysis will ask you whether you want to do sensor data collection, data sharing, or manual entry. So what I'm going to do here is if you don't have it connected by USB, what you can do is then select sensor data collection. You want to make sure that the little red LED is flashing. And then it should be searching for devices. And pretty soon here, the temperature probe will pop up. Oh, hi, Heidi. It's not popped up yet. So what I'm gonna do is I'm gonna put it right next to the computer and try proximity connect. And there, it found it. Oh, it did find it. Oh, there we go. Now it sees it. So what you can see right here is on the back end of the sensor. See if this will autofocus for me. You can see that right here, right there, is the number that is associated with the probe. So right there, it's got this 01 on it. Up, it disappeared again. There it is. So Q1. So I can connect to it. And there we go. So now I'm connected wirelessly. So see, no cable. I'm connected wirelessly to graphical analysis. If I hit collect, I can grab onto the temperature probe and temperature will go up. All right. So just to reiterate, let me go back to the presentation here. So just to reiterate, the simplest way to connect any GoDirect sensor is going to be using the USB port. So here I've got the sensor. You can see the USB ports right here. It's going to be on the end of most sensors like that, most wand style sensors. If you have a phone or you want to use it wirelessly, you can do that as well, just by doing what I just did there, just by connecting it over Bluetooth. And like I said, you can do the same thing on LabQuest app. You can plug it in over USB, or you can use it over Bluetooth. All right, questions. So let me move over here. Now, since most of you are interested in sensors, what I wanted to do was talk about what I think are the key, what we call 4 big sensors for biology and environmental science. And these are the ones that I find most useful in all of the teaching that I've done and training that I've done. And those are the GoDirect gas pressure sensor, the GoDirect CO2 sensor, GoDirect O2, and then the GoDirect optical dissolved oxygen probe. And I'm not going to go through them in exactly this order. I'm going to focus on the CO2 sensor first and then move through them. And I just want to show you some really key features about them. And if there's any features that I don't cover while I talk about them, please let me know. All right, so the big one that everyone usually gets really excited about is our CO2 gas sensor. I just wanted to remind you that the CO2 gas sensor is for gas. You don't put it in water. It is not a dissolved CO2 sensor. And it's a great workhorse. It's great for a lot of experiments. It's terrific for cellular respiration, like say with peas. You can also look at animal respiration with it as well. It's great for photosynthesis. If you wanted to look at how leaves, like spinach leaves, can suck up CO2 or produce CO2 because plants will actually also respire, it's great for that, better than the oxygen gas sensor. Um, you can look at yeast metabolism. So if you want to look at different types of yeast and how they consume sugars or can't consume sugars, it's great for that as well. And then it's also great for soil respiration and then other just basic environmental science applications, like just looking at the amount of CO2 is in the atmosphere, air, or wherever you're at. It's a really, really useful sensor. It's a workhorse. It's one of those that I think every biology teacher could really use, and there's a tremendous amount of experiments that you can do with it. Now, one of the great things about the Direct CO2 gas sensor is it's a multi-channel sensor. So it can measure CO2 gas, but it also can measure relative humidity, and it also will measure air temperature. So it's actually three sensors in one. Now, a lot of people ask us sometimes, why is the CO2 gas sensor more affordable than the CO2 BTA one or the old analog one? That's because since the sensor is digital, we save a lot when we make it, and we've passed that savings on to the teachers or to you or whoever wants to buy it. So it actually is just as accurate and in many ways better than the analog version. The other thing that I didn't say right here is that's actually kind of important is the Direct CO2 gas sensor is also humidity protected. So the sensor itself, the column inside of it where these holes are located right here, there's a little IR column and there's a casing around it that protects it from getting too wet. And it's also temperature compensated, which is something that the other sensor doesn't have. So it's going to compensate for changes in temperature. It's probably one of my favorite sensors. So let's just do a quick live demo for those of you that haven't seen it before. I'm just want to show you how it works here. Let's launch back to graphical analysis. I'm going to widen this up here. Let's disconnect that sensor there. Let's do file new. And I'm just going to connect the CO2 gas sensor over USB. There we go. And it should be that easy. See how easy it was? It just popped right up, just reading around, reading CO2. The y-axis says CO2. It's all ready to go. And if I hit the collect button here, actually, let me just do a little quick experiment just because it's fun. Hold on. All right, so here's the CO2 gas sensor. It's plugged in over USB. I have our little Nalgene bottle right here. I'm going to take this Nalgene bottle and I'm going to put some respiring peas in there. There, so there's my peas. I'm going to stick the CO2 gas sensor in there. And then I'm going to do my little magic trick with the cap, which is put it right there. So that holds it in place. Then I'm just going to wait here for a minute. And you can see that the CO2 level has already gone up a bit. Just going to wait a little bit more, and then I'll hit the collect button. There we go. So you can measure cellular respiration from peas. You can measure respiration from yeast. There's lots of great labs that can be used with this sensor. And like I also said, it's a multi-channel sensor. Let's just wait for the CO2 levels to go up here a little bit. We're waiting there. I'm just gonna let this go up here. I'm gonna clip on or tap on sensor setup. And if you go to the sensor channels, what you can see here is CO2 is not automatically turned. I mean, CO2 is automatically turned on. You could activate temperature and humidity if you wanted to, and then it would show all three. And there you go. It's beginning to go up. And then Yes, yes, you can change the time intervals that it's collected. Let me do that here next. So one of the tricks I wanted to show you as a former biology teacher is students can get kind of bored waiting for this to go up. I know it should be going up, but if you look right here, you can tell that it's already gone up. There's this great little trick here. You just hit zoom to all data, and then you can see that the CO2 level is going up. All right, so question, can you change the time? Yes, let's do new experiment, don't save. If I wanted to change the duration, I would go here to the bottom left corner and tap on data collection settings. And then I can change the length of the data collection or the duration. And I can also change the interval. We have it set to measure at 1 sample every 2 seconds. But yeah, I could change this to 300 or 200. There's a lot of variability there that you can use. All right, let's see. Yes, that's a great thing there. The CO2 gas sensor is a great one for ocean acidification. And yeah, if you want to measure temperature, you can as well. Just so you see that again, all you have to do is tap on the bottom right corner, open up sensor channels, and then turn on temperature and even relative humidity. And now let's say you don't even want to collect data as a function of time. There's also this other really cool feature is you can just turn on larger meters, and I could turn the graph off. So if you wanted to, you can also see the data just as a meter. So that's another key feature of the software. And you can see it's about 25.3 degrees, 25.4, a little higher relative humidity. One of the things just to be aware of with the CO2 sensor and the temperature is the temperature probe for this sensor is located inside the column. And so the air temperature in there is going to be about 1 to 2 degrees warmer because there's a lamp also inside the column than just true ambient air. So just be aware of that. All right, so I'm going to disconnect. There we go. And let's go back to the presentation. All right, any questions about CO2? Oh, wait, let me do that. That's a great one. All right, so what about using yeast? Okay, let's go back to Visualizer. I'll show you how that works. What's great about the CO2 gas sensor is it's super sensitive. So the way that I look at yeast respiration is you just take a Nalgene bottle, you put the CO2 sensor in it, like that's all that you need to do, and then you stand it upright. And then all you would need to do to put in here is about like 2 mils of yeast, 2 to 3 mils of yeast, and 2 to 3 mils of sugar suspension, and you will get great respiration rates. There's an entire lab that you can use for that. And so you can look at the effect of maltose, sucrose, monosaccharides, disaccharides. If you're interested in that activity, I can show you that. I think also we're going to be even doing a lactose webinar later in the term, like in the spring. If you're interested in that, let me know. For CO2, other organisms, I mean, crickets work really great. Caterpillars work really great. It just depends on what you have access to use. We produce, let me just back up here a little bit. Humans produce really high amounts of CO2. So even one of the things you need to be aware of is just like breathing on the sensor is going to make the CO2 level go up really high. But that's why it's really great for being used with animals or other things like that. So yeast, crickets, caterpillars, etc. I don't know if you could detect oxidation or rust over time, but I bet you could. But not in the sensor itself, but if you wanted to actually look at or detect different types of oxidation over time, you might be able to, although an oxygen gas sensor might be better for that. All right. So let's stop for a minute and let's go back to let's go to my second favorite sensor, if everyone's okay with that. And that is the Go Direct Optical Dissolved Oxygen Sensor. And I love that sensor. Here's the optical dissolved oxygen sensor. And let me show it to you over here. Here's what it looks like. What I love about it is so simple to use. Has anyone here actually used a dissolved oxygen sensor before that isn't optical, that doesn't use optical technology? I remember using ones that actually had a fluid that you had to put in the cap, and then you had to let them warm up, and then also they wound up consuming oxygen. Really, really challenging, really hard to use. I could get them to work, but they were always problematic, or I felt like they could be problematic, not fun. What's great about this sensor is all that you have to do is put it in water and measure. That's what's awesome about it. So once again, it's a Go Direct sensor, USB port on the bottom, or we can turn the Bluetooth on. I'm going to plug it in over USB. There we go. Now, what you should see down here is there's this little flashing red and blue, red and blue, red and blue. That is the fluorescent technology in the cap that is actually sensing the dissolved oxygen level. And then there is this nice little round pin here. That is the temperature compensation. So let's just get back to that really quick. Let me just grab some water. Here is a little BOD bottle, and I'm going to put my sensor in some water. Try not to spill here so you can see it's in some water. Put it over here. And then let's just go back to graphical analysis. And you can see that it should be that simple. I have just put the sensor into some water, and it is actually measuring DO concentration right now. So let me just show you the meter. So there you go. No fancy solution to fill up, nothing else is problematic. You just put it in, let it record data, wait for it to stabilize, and it's recording the DO level. It's really a great sensor for that reason. Alone is just how simple and easy it is to use. So it's great for aquatic photosynthesis. It's great for aquatic respiration. You can use it for BOD, biochemical oxygen demand, and also primary productivity. If you want to see, if you've got a sample of water that you want to look at, it's a great water quality sensor, and it's just tremendous for environmental science. To talk about the sensor a little bit, just like the CO2 gas sensor, the dissolved oxygen sensor, the Go Direct optical dissolved oxygen sensor, is also a multi-channel sensor. So it doesn't just measure DO, it can measure DO in milligrams per liter. It can measure percent saturation. It also has temperature, because it's temperature compensated. It can do that. It has salinity correction, and I believe, I may have missed that, it even has barometric pressure. So what we can do is let's go over here. And once again, if we tap on the bottom right corner where it says sensor setup, we can look at all of the channels here. And yeah, and you can see there's pressure, there's a salinity. So if you know the salinity, you can add that in and it'll correct for salinity, temperature, and saturation. So you can actually look at DO concentration and saturation at the same time if you wanted to, which is great. All right, calibration. Someone asked about calibration. That is great. What I wanted to let you know is that both of these sensors, the CO2 sensor, the Go Direct CO2 gas sensor, the Go Direct optical dissolved oxygen sensor, and yes, even the Go Direct oxygen gas sensor are all calibrated in software, and it's a really easy calibration process. So if you want to see that, I can show you how easy that is to do. Let's go to Visualizer. You would normally put the sensor in the little bottle that it came with right here. You would want it to be standing up, right, for 60 seconds. What I have in here is I have a damp sponge. So in this little bottle, there's some water in the sponge. I place it in the air above it. So we know there that it's in a 100% saturated environment. And then all I have to do is go over here and tap on DO concentration, hit calibrate. Select calibrate now. And there we go, it's calibrated. The CO2 sensor calibration is done in the same way. It's really simple. You just take the CO2 gas sensor, you want to capture a sample of air from outside. Hold on here, let me move this away for really quick. Actually, there we go. You want to capture a sample of air from outside, and then all that you need to do is do the same thing. Tap on where it says CO2, select calibration, and what it'll do is it'll tell the sensor, hey, I'm at 400 ppm, and it'll say that's 400 ppm. So it's all done in software. The oxygen gas sensor, same way. You just calibrate it to outside air, which would be around 21%. I can take you through more on how to do that, or we can talk more about it. Or if you want to know more specifics, you can always email me at biology@vernier.com and we can talk more about it. All right, someone asked, are these sensors water resistant or waterproof? That's a really great question. Like I said, the CO2 gas sensor is humidity resistant, but it is quote unquote not waterproof. But it's resistant to high humidity. It's made as a gas sensor, so it's not made to be dunked in water. The oxygen gas sensor is not as water resistant, but it'll work in relatively high humidity environments. This sensor right here, the DO probe, is water resistant. It's a great sensor for that purpose, that it is water resistant. It's actually one of them that's pretty close to waterproof. As far as how much water needs to be in the sponge, it's not crucial as long as the sponge is wet. Like you want to make sure that the sponge is nice and wet. You can just make sure there's about 1 centimeter of water in there. You just want to make sure that the tip of the sensor is in the air and that it's really nice and humidified so that it's at 100% saturation. Okay, I've got another question, which is, will brackish or salt water affect them even in air? Well, the only sensor that you would probably want to use in brackish water would be this one, right? The DO sensor, and that's what the salinity calibration is for. You can offset it. If you know the salinity of the solution that you're measuring, you can put in the salinity and it will automatically correct for that. All right, let's go back to the presentation here. Other questions? These are all great presentations, great questions, by the way. Thank you so much for asking. All right, and I did the live demo. All right, so that's a good question. Someone just asked, a teacher stored one of the, stored a sensor in water and it doesn't work. What should you do? The best thing to do is to email us at biology@vernier.com, contact us over chat, or just call in. Let's go to the website really quick. So what you can do is you can go to support, and there's lots of videos here, but you can email us or you can call us. And then I think there should even be a chat window here that would open up. You could even chat with us and just ask us what's going on. But almost all of these sensors come with a conditional five-year warranty. So the GoDirect DO sensor should be okay as long as the tip was just put in water. We would definitely take a look at it and we would try to fix it. There might be some intrusion into it, but we still would try to fix it. It might be that the cap just needs to be replaced. All right, other questions? Yeah, the biggest answer to almost any question, if there's an issue with my sensor or I need a lab or I've got another question, is just get in touch. There's 3 key people in the biology department that would answer your question. That would be me, John Melville, Sarah Talarovic, or Colleen McDaniel. And that's why we often say, hey, email us at biology@vernier.com, because if you do that, it's going to go directly to one of the 3 of us, and you'll get a biologist straight away. Oh, great question. The question was, do the GoDirect sensors contain batteries? Yes, they do. They do contain batteries. They contain a small lithium-ion battery that some are bigger than others. They can be replaced. So you can replace them or you can send them in to us and we can replace them. But there are batteries. That's actually what powers up the Bluetooth, etc. So yes, they do contain small lithium-ion batteries. Other questions about DO? Okay, another question. The stations are in recall. Some of them are. We're getting that fixed. They're still great things. They're going to be great to use. Another thing that you can do though is you can also buy a powered hub. That's another option to charge your sensors as well. Someone had, let's see, Carissa had the question, well, the old LabQuest won't work with these, correct? It depends on which one. So do you have an original LabQuest that's teal blue? Do you have a gray LabQuest that is a LabQuest 2, or do you have a black one, which is a LabQuest 3? They will work with LabQuest 2s. So Carissa, and they will work with LabQuest 2s over USB. The question is, will the Bluetooth work? You may have to update the software. And if you go to our website once again, and go to, let's see, go to downloads. If you go to downloads, you'll see the companion apps, but then if you go down, you will see software updates. And you can go right here to your LabQuest 2, and you can apply the LabQuest 2.9 update, and you can see that resolved issues with some GoDirect sensors, added support for Go Motion and CBR2, and then it will give you instructions on how to install that LabQuest 2 update as well. It says to use a USB flash drive. I recommend using an SD card if you can. It'll be a lot faster. That's what we use here. And there is an SD card port on the LabQuest 2. All right, another question someone had before we move on is for the DO probe. When putting it in a stream, which of course is colder than the air, how long does it take to equilibrate? I would say give it around 60 to 90 seconds. Just let it sit there for 60 or 90 seconds, and that should be about a good period of time. Another question was, what is the range? Great, and then field question. So let's just talk about the range really quick. So let's just go back to Vernier here. If you want to check the accuracy settings or anything like that on the sensor, all you have to do is find the sensor on our website. If you just type in the order code or the product name, I just know the order codes by heart. Here's the GoDirect optical dissolved oxygen probe. And if you scroll down, you can go to the specifications and it'll show you the range. It's 0 to 20 milligrams per liter or 0 to 300%. And it'll give you the accuracy. It's plus or minus 2 milligrams per liter, below 10 milligrams per liter, or plus or minus 0.4 above that. And then in the percent saturation, it's plus or minus 2% below 100% and plus or minus 5% above 100%. If you want to talk about using them in the field, that is a great question. You can totally use them in the field. They're great for field work. We do several workshops with people in the field. That is a really great reason to get a LabQuest 2, or if you have a LabQuest 2 or a LabQuest 3, and that's because LabQuest 2 and LabQuest 3, if you go to sensor setup, they have GPS. So you can actually turn on the global positioning system and you can actually collect data while collecting your satellite lat and longitude and stuff like that. It's great. That's probably a little bit much for the time that we have here, but if you're interested in that, like how to use them for field work, please just email me at biology@vernier.com. We can do a little webinar for you. That's another great thing to add is that everything that I'm doing right now in this hour, if you wanted a personal webinar with one of us, me, Sarah, or Colleen, we would do it for free over Zoom as long as it's under an hour. So just remember that if you need training, online training, etc., it's going to be free as long as it's under an hour, and we will help you out with your sensors or talk to you about other sensors as well. All right, let me ask Someone asked about accuracy. That's a great question. But it's for pH. You can use a mixture of these. What I would recommend is to just look at the specifications on the website for each sensor for like pH, go direct pH and the wired pH, and just look at the specifications right here. It's going to be on the tab. And what I would recommend is if you're definitely doing this for research, you should definitely calibrate them, right? Calibrate them before you use them. And that's going to give you the best accuracy that you can hope for. And I found they work great. Cool. All right, someone asked, are there sensors that can be used in the classroom and then returned? We do not have a loaner program. That's a great idea, but we do not, but I bet you lots of other schools do or other places. If you're looking to purchase and you're thinking about a large purchase, you can ask for a preview and we will find 1 or 2 sensors to send to you. Once again, if that's something that you're interested in, just email me at biology@vernier.com and we'll take care of that. Are there any other sensors that I would recommend for aquaponics? There are. Email me at biology@vernier.com and I'll let you know what they are. The PAR sensor is really great. That can be used for measuring light levels underwater. It's terrific. We have other probes that can be used for field work. I'd love to just do a whole webinar just on sensors that can be used for field work. Let's get back to the last 2 sensors here. I cannot review right now how to calibrate the go direct pH sensors, but I'll show you where you can. Because one of the great things that you can do is you can go to our YouTube channel and there's going to be videos there of how to do a lot of what I'm telling you what to do. It's a great resource. All right. So let's just talk really quick about the go direct O2 gas sensor. And once again, thank you for so many questions in chat. This is really terrific. I love how engaged you all are. So feel free to keep those coming. So the go direct O2 gas sensor, it's really useful for enzyme action. It's great for photosynthesis. It's great for cellular respiration, and it's great for animal respiration. I put some of these with little asterisks by them because I really think they're great when you use them with the CO2 gas sensor. There are a few tricks that you need to know to get them to work in the cases for photosynthesis and cellular respiration, but I can explain to you those really quick. Let's just take a look at these side by side again. The standard lab for looking at cellular respiration with peas says to use 20. If you want the O2 gas sensor to work really well, I recommend filling the entire bottom with peas. And then I also recommend using our BC 250 or our biochamber 250. So it's got a port here. And the oxygen gas sensor right here needs to stay upright. So you can pop it in here into the chamber. I'm going to pour my peas in here. And then you can place the CO2 gas sensor in there as well. And there you go. Now you're ready to measure CO2 and O2 at the same time. If you're interested in measuring photosynthesis, spinach leaves work great. The big key thing that we found though is that you need to make sure that you get the right light source. And there are really good LED light sources on the market that can be used for that. That will work great. The oxygen gas sensor should be calibrated before use. The oxygen gas sensor is really simple to calibrate. We can go over that really quick. Let's just go back to Vernier Graphical Analysis. I am just going to grab an O2 gas sensor, connect it. And you can see it's already reading about 20.5%. That's because I calibrated before I used it. But it's really simple to calibrate. You want to just make sure it's an open air, which should be at around 21%. Go to calibrate. And then what's great is it's going to tell you when it was last calibrated. It was calibrated today. And then this relative stability reading is going to tell you like how much it's degraded. It's at 86% of what it was originally. So it's degraded a bit. but not much over several years. And what I can do is just tell it, hey, 21. I want you to read 21, hit keep, and apply, and now it's going to be reading 21. We worked really hard to make it so that all of these Go Direct sensors now are all calibrated in the same way. It's a one-stop, click on the meter, calibrate it. So it should be a lot easier for any biology teacher to calibrate the biology sensors or environmental science sensors. Great. Other questions about O2 gas? Gotcha. So that's a good question. If you wanted to measure, so Christy asked, what about measuring two sensors at the same time? Well, there's a couple ways to do that. You could just connect them both to graphical analysis. So right here, I've got the O2 gas sensor attached. So I've got the O2 gas sensor attached by USB cable. Let's turn on the CO2 gas sensor. I could either connect it over USB, which would be the simplest, or it's just searching here for the CO2 gas sensor. Let's see if it finds it. I could connect it over Bluetooth, and then it would collect them together at the same time. While we're waiting for that, Judy Day asked a great question, which is, how should the O2 gas sensor be stored? It needs to be stored upright. So there's an electrochemical cell in the base here, and that electrochemical cell has fluid in it. That's part of the reaction that measures the O2 gas. And you want to make sure that it's stored upright. So one of the things we say is just, you can pop it in the bottle that it came with like this and store it upright. That's also the reason why it comes in a bag when you get it out. So you wrap it around the bag, wrap the bag around the sensor itself, and then you can pop it on top of the sensor. And because you want to limit the amount of oxygen that the sensor is exposed to itself. Okay, there we go. CO2 sensor is available. I'm going to hit connect. Up, it lost it there. Let me just stop there for a minute. We'll go back. No, so the CO2 gas sensor is great. It can just be stored dry. It's solid state. It's a great sensor. It can be stored upright, sideways, however you want. That's why I really love it, because it's one of those that you don't have to worry about how you store it. You don't have to really worry about making sure the membrane is wet like the dissolved oxygen sensor. It's a great sensor and is a terrific one to get started with if you just want to start using Go Direct probes in your classroom. All right. So the gas pressure sensor is just terrific because you can use it for almost everything. You can use it for grip strength, enzyme action, transpiration, fermentation. It can be used for just about everything. So let me just show you that really quick. And then we'll stop for some questions. Here's a visualizer. This is the software that's used to view my little camera. The Go Direct gas pressure sensor has a little lure lock here, and you can use it with a tube and a little bulb to measure like grip strength. It also comes with a little key kit of accessories. So you can actually attach the gas pressure sensor to a test tube. Right, so if you had I've got a little 50 millimeter conical tube, you can pop it in there and just twist it tight, and then you can measure gas pressure changes. It comes with another stopper, this one right here, that can be used with a flask. And that's one of the other great things I think about Vernier is we try to give you all of the parts, components, and pieces you would need to do an experiment. So the CO2 gas sensor, right, is going to come with an Nalgene bottle. The O2 gas sensor is going to come with a Nalgene bottle. The gas pressure sensor is going to come with all of the little gadgets that you would need to use it for a variety of labs. All right. So what about labs? I'm just wondering if all of you know Oh, someone asked if the oxygen gas sensor is stored on its side, will it still work? Best thing to do is just pop it up, store it upright, and see if it responds. Since you're asking all of these great questions, let's just try and find Vernier YouTube. And you see the first thing that pops up is Vernier Science Education. And there's going to be all of these great resources here that you can use. You can search a lot of the cheat sheets that you want on how to calibrate sensors and stuff. It's all going to be located right here. This is a tremendous resource that we now have that anybody can get to just by going to our YouTube channel. Now, what about labs? If you have some of our old lab books, you can still get updated lab instructions. So let's say you have Biology with Vernier. This is the new book, Biology with Vernier, 4th edition. Maybe you have the old book. That's okay. A lot of the instructions in it have been updated for graphical analysis. And you can get access to it. You just have to go to our website, look in the top right corner, select login and register. And then you can either get to your account or register for an account. And if you already have Biology with Vernier or a book, all of the updated contents will be located in there. So the big thing to do is to create an account and make sure that you use your school email address when you create that account. If you need personalized training, like I said, we'll give you personalized training. You can also tap on here where it says educator training. There's personalized training that you can get for free. If it's under an hour, someone could come out to as well. And then if you want this webinar, look at other webinars, just tap on where it says webinars and you can take a look there. All right, we've answered a lot of questions and we've got like 7 minutes left. And I think go ahead and ask some questions here, but if you have some time, Dylan would like you to fill out the survey and then just feel free to ask me any other questions in the time that we have left. Thank you so much for this engaging webinar. This was a lot of fun. So any other questions?
While folks are thinking of their questions, we could go ahead and select the winner.
Yay.
Amazing. So I have a random name generator here. I put all of the attendees' names in there, so I'm going to go ahead and let it select the winner for us. So I will go ahead and spin the wheel. It's thinking. And it looks like Christy B has won the Go Direct temperature probe. Congratulations, Christy.
Yay.
Amazing. So I have, yes, yay. I have your email information from your webinar registration. So I'm just going to email you a link to our forms so that we can make sure we have the right information for you. And congratulations.
Great. And then I think let me see if I can, it was even a webinar here. I think an older webinar that even had some stuff on how to actually store your sensors and stuff like that. So that was an entire webinar that was done over the summer. Go ahead and look at under our webinars or look at the YouTube video and you can find out all about that as well. So you can just browse past webinars here and there's a ton of webinars that you can look up. Thank you so much for coming. This was a great webinar. Really appreciate it.
Well, if we don't have any questions, we could go ahead and wrap. Does anyone have any burning questions they need to get out while we're still on the webinar here?
I think doing a field-based webinar would be terrific. Hopefully we can do something like that. Oh, here it is. Here's the sensor calibration and maintenance tips and tricks with Vernier and Case. That'll have a bunch of sensor calibration and maintenance tips right there. So that would be a great webinar to go watch. All right. Thank you all for coming.
Thanks, everybody.